Hi, welcome back to Bioinformatics Introduction and Methods. Let's continue to learn about bioinformatics resources. Let's move from the US to Europe to look at resources at the European Bioinformatics Institute, or EBI. The European Bioinformatics Institute was established in 1994 and is a part of the European Molecular Biology Laboratory, EMBL. It is located on the Wellcome Trust Genome Campus in Hinxton, near Cambridge in the UK, and shares a campus with the Wellcome Trust Sanger Institute. It has over 500 staff, including PIs, postdocs, students, software developers, data curators, etc. EBI has resources for DNA, RNA, and protein sequence retrieval and analysis, protein structure analysis, gene expression analysis, pathway analysis, ontologies, and so on. For genes and genomes, an important resource at EBI is the Ensemble database, that is a comprehensive secondary database of gene and genome annotations. EBI also archives raw sequence data such as metagenomics data. It is also home to the 1000 Genome Project that we talked about earlier in this MOOC. EBI supports blast search of sequence databases and multiple sequence alignment by cluster omega. For protein sequences and functional domain annotations, EBI provides several very useful secondary databases, including UniProt, InterPro, and PFAM. In addition, it hosts PRIDE, which is a large repository of mass spec proteomics data. EBI also provides data on protein structures in resources such as PDBE, PDBSum, and quaternary structure. In addition to databases, EBI also provides lots of tools for protein sequence and structure analysis, such as InterProScan, PDBE PISA, ProFunc, and PDBE Fold. In addition, EBI is home to expression data repositories such as ArrayExpress and Expression Atlas. Another area of strength of EBI is its resources on reactions, pathways, and ontologies, such as the INTERACT database of molecular interactions, Reactome database of pathways, and biomodels of dynamic network models. EBI's enzyme portal and catalytic site atlas have annotations of lots of enzymes. Small molecules can be found at CHEBI and CHMBL. Metabolomic data can be found at Metallolite. Ontologies include the gene ontology, which we had talked about at length in earlier lectures, as well as systems biology ontology and experimental factor ontology. Search engines for ontologies include Ontology Lookup Service and QuickGo. Let's now look at some of the resources in a little more detail. Ensemble provides comprehensive genome databases that incorporate many types of data and annotations in addition to the genomic sequences, including gene expression data, genetic variations, cross-species conservations, etc. Ensemble includes lots of vertebrates and other eukaryotic species. For the human genome, the current Ensemble version, GRCH37, contains 3.3 billion base pairs, 20,000 coding genes, 9,000 short non-coding genes, 13,000 long non-coding genes, 14,000 pseudogenes, about 200,000 transcripts, 55 million short genetic and somatic variants such as SNPs and indels, and 10 million larger structural variations. There are 4 types of data in Ensemble. Curated data are manually curated based on experimental results and validations. They serve as the reference for future studies. Large-scale projects contain data that are not curated but are produced by high-throughput experiments. They tend to have a large sample size or data volume. Other studies include uncurated low-throughput experimental data, possibly with some computational post-processing. Finally, only computationally analyzed include secondary data that are generated by running automated algorithms over curated or reference data and large-scale high-throughput data such as the genomic sequences. Although they don't have as high a quality as curated data, they cover a lot more genes and can save you the trouble of running the algorithms yourself, as many of the algorithms are time and resource consuming. Another useful resource at EBI is the protein knowledge base called UniProt-KB. UniProt-KB has two parts. The first part is the manually curated SwissProt. SwissProt currently contains curated protein annotations for 541,000 entries. The second part of UniProt-KB is Tremble, which contains over 48 million entries of automatically generated, uncurated annotations of proteins. As you can imagine, the quality of Tremble is not as good as SwissProt, but it covers almost 100 times more proteins. You may remember from the last slide that the growth of the number of entries in SwissProt had slowed down since 2010. But as you can see from this slide, the growth of the number of entries in Tremble continues to be very steep. My guess is that the growth of the number of new proteins had far exceeded the capacity of EBI to annotate them manually. A useful resource for molecular interaction is INTERACT. It currently contains over 436,000 interactions involving 80,000 molecules based on data from 31,000 experiments published in 12,000 papers. As you can see from these two figures, the amount of data in INTERACT had been increasing steadily over the years. The interactions in INTERACT were determined by experimental technologies such as tandem affinity purification to hybrid pull-down and so on. The most common type of interaction is physical association. Other types of association are also common. Other interactions include colocalization, phosphorylation, and dephosphorylation reactions, and so on. The species with the most data available are human, yeast, and mouse, followed by fruit fly, C. elegans, E. coli, and Arabidopsis. This is probably not surprising, as these are some of the most widely studied species. A useful tool maintained by EBI is Cluster Omega for multiple sequence alignment. It has a nice web-based user interface that is fairly easy to use. A related tool is Cluster W2 for phylogeny. It can generate phylogenetic trees given a set of input sequences based on the Cluster W2 algorithm. Another two very useful related resources are Interpro, which is an integrated secondary database of protein families, domains, and motifs, and Interpro Scan, which uses the data in Interpro to predict the functional domains and sites in your new proteins of interest. When I was doing my PhD at Stanford, I used to have to go to several different protein domain databases in order to get a comprehensive prediction of my proteins of interest, as different domain databases and prediction methods had different focuses and strengths. Today, Interpro integrates most of the protein domain databases and prediction methods in one unified interface, making it very efficient and convenient for users. Last but not least, the proteomics identification database, PRIDE, is a large, centralized, standardized repository for proteomics data, including protein and peptide identification from mass spec, post-translational modification from mass spec, and the supporting spectral evidence. In the next unit, let's look at another centralized resource, the UC Santa Cruz Genome Browser. See you then.